Why are we here today? What are we doing? Why the state of biodiversity? Because the state of biodiversity is important. The intrinsic value of biodiversity aside, there are undeniable benefits of biodiversity and healthy ecosystems to human health and quality of life. So why the state of biodiversity? Because we think everyone should have the knowledge to be strong advocates for the natural world and their own quality of life. And as conservation and advocates for sustainable land stewardship, the San Diego Natural History Museum recognizes and respects Indigenous peoples as traditional stewards of the land. Specifically, we recognize the Kumeyaay people whose ancestral homelands the museum currently occupies. We extend our respect and gratitude to the Indigenous people who have lived on and cared for this land since time immemorial. As the original caretakers and conservationists, we honor their continued legacy of understanding, caretaking, and upholding the pillars of biodiversity. So with that, I'm going to now introduce our first speaker, who I'm super pleased to have join us. Dr. Chelsea Wood is an ecologist interested in how environmental change impacts parasite transmission. She received her Ph.D. in 2013 from Stanford University, where she was a student of marine ecologist and conservation biologist Fiorenza Michelli. She went to complete postdocs at the University of Colorado and the University of Michigan. Chelsea is currently assistant professor in the School of Aquatic and Fishery Sciences at the University of Washington in Seattle. She's published 49 scientific papers on biodiversity and infectious diseases, secured over $6 million in grant funding, and is a recipient of numerous early career awards, including the Kavli Fellowship from the U.S. National Academy of Sciences, a Sloan Research Fellowship from the Alfred P. Sloan Foundation, and the Ecological Society of America's Early Career Fellowship. We're very fortunate to have Chelsea with us today. Thank you so much for giving us your time, and I really look forward to your talk. Thanks a bundle.
Thanks so much, Michael. That was a very kind introduction. I appreciate it. It's a big honor to be here today talking to you about the surprising relationship between biodiversity and disease risk. I got curious about this question a couple of years ago, and with the help of a few colleagues, I undertook a massive data analysis project to find the answer. And if you want the nitty-gritty details, you can find them in this publication. I'm going to drop the link to the paper in the chat at the end of the talk, but for now, you'll see that I've actually redacted a couple of words from the title, and that's because they give the punchline away. Instead of spilling the beans right up front, I want to hold you guys in suspense for the next 10 minutes. When I was first getting interested in this topic years ago, I had a favorite Sunday morning ritual. And let's see if I can advance this. There we go. I would sit down to coffee with an actual paper copy of the New York Times. And one sunny Sunday, I opened up my paper and I came across an article with this title: "Destroying Nature Unleashes Infectious Disease." That an article like this one ended up in the New York Times really says something. This is an idea that's become very influential in the disease ecology world, and it's now even made it out beyond the confines of the ivory tower. So the formal name for this hypothesis is the dilution effect. And the dilution effect hypothesis was originally developed to explain patterns in Lyme disease transmission. And Lyme, of course, is caused by a bacterium that's transmitted to people in the bite of ticks. And the idea of the dilution effect is that a forest that is intact and unimpacted is going to have a super diverse mammalian fauna. Meanwhile, a degraded habitat, for example, where development for housing has happened, is going to have only a small subset of those mammal species. Now, Lyme doesn't just grow in humans, it can also grow in other mammals, but not every mammal is equally good as a host for Lyme disease. Mice tend to be really good hosts for Lyme, and it just so happens that mice are also the mammal species that does best in impacted, degraded forests. So when a new generation of ticks emerge, their meals are really different in unimpacted compared to degraded forests. In unimpacted forests like the one on the left, only a small fraction of the ticks feed on a Lyme competent host. So if you then go for a walk through that unimpacted forest and you get bitten by a tick, there's only a small likelihood that that tick is going to be carrying Lyme disease and that it will transmit Lyme to you. In contrast, if you get bitten by a tick in a degraded forest, that tick is highly likely to carry the Lyme disease bacterium. Now, we know that this probably happens for Lyme disease, but many have argued that this may be the way that disease works generally. The dilution effect is even sometimes called a law of disease ecology. And whether you believe that or not, it's certainly the case that our literature has become very focused on this hypothesis. And for good reason, right? The idea is super appealing. It means that conservation and public health can go hand in hand. It means that we as ecologists share common goals with the Centers for Disease Control and the World Health Organization. And most importantly, if biodiversity protects us against infectious disease, it means that we have this real visceral incentive to conserve it, something that's going to convince even the most hard-hearted person who otherwise would be totally indifferent to conservation. But I wondered something about the dilution effect. Sure, it works in Lyme, but all ecosystems contain more than one parasite. What would happen if we looked across the parasite diversity within an ecosystem? Would we see 100% dilution across all of these different parasite species? What's missing is a systematic evaluation of what is actually occurring in nature. Because if conservation dilutes the transmission of one disease and amplifies the transmission of another, then we're in trouble. In order for conservation to be effective as a disease control tool, dilution needs to work for most diseases. It has to be general. Tons of folks have predicted that the dilution effect is likely to be general, and you can read about that in lots of papers in a variety of really well-respected journals. And I think that these papers make what is a very reasonable prediction, that real-world conservation will protect real-life humans against disease. If that's the case, then where nature is destroyed, we should be able to measure an uptick in infectious disease burden among people. And that's what my team and I decided we wanted to test with data from real life humans. To do that, we relied on one amazing resource, the Global Burden of Disease dataset from the Institute for Health Metrics and Evaluation. These are the same friendly folks in Seattle who brought you COVID predictions in the beginning of the pandemic. These guys also maintain a comprehensive database of all the drivers of human morbidity and mortality. We extracted information on the burden of disease for each country and each disease, and then we calculated the change in disease over time from the beginning of the dataset in 1990 to what was then its end in 2010. And we didn't play favorites. We extracted data for the World Health Organization's 24 most important infectious diseases of people. And as you scan through this list, you'll notice a lot of big name diseases. We got malaria, schistosomiasis, dengue, rabies. You'll also notice that Lyme is not on this list. That's because Lyme is not important enough in terms of the total suffering that it causes at a global scale to get tallied by the Institute for Health Metrics and Evaluation. You'll also notice one other familiar character who's missing from this list. These data come from the before times. So remember, as we're going through the results of this study, we are just talking about the global disease landscape as it was before COVID-19. Okay, here's where we get to the punchline. After all this number crunching, what did we find? Let me start by setting up this plot for you. On our horizontal or x-axis, you're going to see all the factors that we were interested in. You'll see that I've got biodiversity, the variable that we were most interested in, all the way down at the end on the right, alongside forestation, which is another proxy for how much conservation a particular country is doing. But we've also got the other factors that we wanted to include, like urbanization, wealth, and population density, as well as temperature and rainfall, which are two components of climate change. We wanted to include these variables because we know that they're probably important in governing disease transmission, and we wanted to see how biodiversity and forestation would stack up with these other predictors in terms of their importance. So that's the x-axis. And then on the vertical or the y-axis, you're going to see a value that tells us how does disease burden change in response to increases in each factor. So if disease burden increases with increasing urbanization, it'll fall above that dashed line that indicates 0. And if disease burden decreases with increasing urbanization, it'll fall below that dashed line that indicates 0. Okay, now I'm going to throw the actual results up on the plot. We'll start with urbanization. It looks like countries that urbanized between 1990 and 2010 tended to see a greater reduction in disease burden than countries that didn't do as much urbanizing. That's weird, right? I think of cities as cesspits of disease. No offense, Seattle. I love living here, but that is the association that I had in my mind until I did this analysis. And I puzzled over this result for a while until I realized that cities aren't just a bunch of people crammed cheek by jowl. Cities are places where a doctor is always close at hand, where it's cheaper and easier for the authorities to provide clean piped water and sanitation, where public health agencies can efficiently reach a lot of people fast for education or vaccination. This result suggests that cities are actually good for people's health. Okay, so one surprising result. Now let's look at wealth. The error bar for wealth overlaps 0, which means that we don't know for sure that it's different from 0. So we see this trend for countries that get richer over time to see greater reductions in disease burden than countries that get poorer over time, which makes sense, but the pattern is not statistically significant. No significant effect of population density, no clear effect of temperature, and no effect of rainfall. So for our 2 climate change variables, temperature and rainfall, it doesn't look like there's a consistent effect across all the 24 infectious diseases that we tracked. Okay, here's where things get weird. Countries that increased in forestation over time actually seemed to increase in their disease burden. That's weird because it's exactly the opposite of what the dilution effect would predict. And then finally, the answer that everyone's been waiting for, there is no coherent role of biodiversity in disease burden in this data set. Our error bars overlap 0, so it doesn't look like changing the biodiversity of your country has any impact on disease burden in that country. So what do I want you to take home from this? Well, first we have some good news, right? Urbanization appears to offer pretty strong protection against the infectious diseases that we tracked. And that's actually great news in a world that's rapidly urbanizing. But of course, there's also bad news. Biodiversity does not appear to offer a protective effect for the infectious diseases that we tracked. And in fact, there's this worrying signal that increasing forestation might actually increase disease burdens, which is exactly the opposite of what the dilution effect predicts. So what's going on here? Well, forests are sources of lots of disease. You do not get malaria sitting in an air-conditioned apartment. So while it's possible that conservation might be a useful disease control tool for a handful of individual diseases like Lyme, we need to be aware that it's equally likely to exacerbate the transmission of other diseases. We have to be able to anticipate and avoid that, because the absolute last thing that we want to do is a conservation project that gets people sick. There has been so much hope in the disease and community ecology literature that biodiversity might yield this consistent ecosystem service of disease control. But our work suggests that that hope is unwarranted, at least for the 24 most burdensome human infectious diseases that we tracked. Instead of insisting on win-wins for conservation and disease, I think we need to be looking for targeted opportunities to do each of those things well, separately. Please do not take what you've learned here today and use it as justification to cut down a forest. Ecosystems offer many services, but control of infectious diseases is probably not one of them. And that's because human infectious disease burdens decrease with urbanization, but they do not decrease with increasing biodiversity. Thank you.
Man, Chelsea, that was awesome. I feel like you could have just done a mic drop right at the very end of that. That was so super well done. Such clear messaging, and I really appreciated the way that you set up that graph to help people understand the results. So thanks for taking the time to put that together. I do have one question for you, in because when the COVID stuff was first coming out and people were talking about pangolins versus bats and all this sort of stuff. I was seeing papers specifically about the shedding, particularly related to bats, with the shedding of viruses being on the uptick, associated with like forest destruction and things like that. And I know that we're not necessarily talking about a parasite here, but are there other areas of human human disease biodiversity interaction where we do see that destruction of biodiversity is increasing the risk of exposure in some sort of way?
Yeah, that's a really interesting question. So first, I want to make a distinction between the diseases that we tracked in this study and things like COVID-19.
Yeah.
The diseases that we tracked are well-established human infectious diseases that have evolved alongside humans for a long, long time. They're nothing new to humans.
Gotcha.
So we understand their transmission, and their transmission works a little bit differently than the dynamics of spillover that happen at that frontier between wildlife and people. That said, we can learn some stuff from the relationship between biodiversity and established human infectious diseases that we can use to understand these diseases that are just now spilling over into the human population from bats, the way that, for example, COVID-19 did, or SARS, or Ebola in 2013. And one of the things that we can learn is that contact with biodiversity is the most important predictor of whether spillover can happen. Spillover is probably occurring all the time, every day, from human wildlife contact. And you have to have a huge number of spillover events in order to get that one that is effective at human to human transmission. If you guys have ever seen the movie Contagion, you know how that process works. And so I think you can kind of look at that problem from either perspective, right? There's some really good evidence to suggest that there is more viral shedding among bats in these frontier regions where forest is being destroyed. But you also have to have that biodiversity in order to have that pool of pathogens that spill over into people. And so that's why I conclude with, this is not an excuse to cut down forests, right? Technically, if we mowed down all forests, we would never have another COVID-19 event, but we would also all die, right? Forests have lots of ecosystem services that they offer other than these ones that we posit about infectious disease control. I think the key is to separate humans from wildlife, right? To reduce the opportunities for things to spill over from animals into people. I think conservation is a really worthwhile thing. I'm an ecologist. I got into this business because I love nature, but I'm just not sure that we should use it as a disease control tool for established human infectious diseases or for these emerging infectious diseases.
It's fantastic. Chelsea, thank you so much. I was really looking forward to your talk because I had read some of your work, and I love stuff that subverts our expectations, and fabulous work. And we'll have more time for Q&A with Chelsea later in the panel discussion. For now, we are going to move on to our next speaker. And thanks, Chelsea. And our next speaker is Dr. Mrinalini Watsa. She is a scientist in conservation science and wildlife health at San Diego Zoo Wildlife Alliance. Her research focuses on the application of frugal science in the field of conservation genomics in the Peruvian Amazon for wildlife disease and biodiversity monitoring programs. In addition, she works on population genomics monitoring of some of your favorites, like the Andean bear, the Sumatran tiger, Indian and African leopards, and reticulated giraffe. Programs she manages emphasize capacity building and research training and conservation education as the fundamental pillars of sustainable field biology. Thank you for joining us today. I turn it over to you, Mrinalini.
Thank you, Michael. Can you hear me well?
Yeah.
Okay, great. And the slideshow looks okay too? We're
beautiful.
Good? Okay, great. Thank you. So thanks very much for the opportunity to speak to you today. Y gracias por las traducciones españolas de esta charla también para ayudar a este mensaje a llegar a un público más amplio. So I'm a scientist at the San Diego Zoo Wildlife Alliance, and today I'd like to discuss the factors facing us as we consider effective pandemic alert systems, including a possible solution of using in situ laboratories to do this. So wildlife biologists are a special group of people. They operate in a field in which A, there is very little money compared to almost anything else connected to human beings. B, the contexts are highly varied and challenging. And C, nothing is ever designed for your species of interest. So if by some miracle it'll cost a small fortune, which brings me back to point A, that is, there is no money. So as a group, we have to think outside the box and adapt. When the pandemic hit, a group of us, ranging from disease ecologists to social scientists to genomicists, formed the Wildlife Disease Surveillance Focus Group to answer one question: how can we make rigorous wildlife disease surveillance practical and more democratic? And I've been invited here today to present the opinion that we published, and you can see the paper down there on the right if you'd like to look at it, and I'll put the link as well in the chat at the end of the talk. So diseases emerge when hosts or pathogen ecology are changed. And despite coronaviruses now becoming somewhat of a household term during the present pandemic, disease outbreaks are not only threats to human populations, and neither are they new. In fact, spillover events are extremely serious threats to endangered species, which already exist in small population sizes, usually with reduced genetic variation. They have disappearing habitat, and this makes them highly vulnerable to extirpation by disease. So for example, black-footed ferrets that were thought to be extinct, then discovered and bred in captivity and reintroduced, are plagued literally by Yersinia pestis. It's a bacterium that has decimated their populations, and it comes to them from rodents. Canine distemper has affected African wild dogs. Ebola has been found in gorillas. And so the list goes on. And over the course of early European human expansions across the globe, diseases have moved where people move, and with them. And it's likely that outbreaks have played a somewhat unrecognized and most likely undocumented role in reshaping populations of humans and wildlife across time. So what do we know? We know that about over 300 emerging infectious diseases, or EIDs, have had an impact on human populations over the past 50 years or so. And although viruses represent only a fraction of the approximately 1,400 known human pathogens, they actually place a disproportionate burden on global health. And a large number of these remain entirely undescribed. But what of the host species? Currently, only less than 0.5% of the mammals and birds on the planet have genomes that have been fully sequenced. And this number would be even lower if you start to think about all vertebrates or broader groups. So how do species interact and rely on each other within ecosystems? What is normal for them? What are, in fact, what we call the baselines? The only thing we really do know is that we don't know enough. So wildlife, humans, and livestock interact globally in an interface that is as intricate as it is large. This interface can never be fully eliminated, and attempting to dismantle it completely would be as unwise and unrealistic as suggesting that we eliminate an entire species just to protect human beings. So there are two commonly held beliefs that I want to point out here that are actually fallacies. One is that we cannot claim that wildlife markets are more or less dangerous overall than farming, particularly when farming animals on the scale at which we do currently, to feed millions of people, is done on that scale, right? So there's very little information that can actually give us a good way to say this is definitely the most dangerous thing. For example, the H1N1 swine flu that killed literally thousands of people in 2009 emerged in North America as the result of reassortment of naturally occurring pig viruses in these domesticated livestock that were being farmed at very high numbers. So the figure on the left is from a paper on the global trends in infectious diseases at the wildlife-livestock interface. The second thing I want to point out here is that this is not a problem that belongs to someone else, caused by someone else. North America and the United States in particular remain a common endpoint for the consumption of wildlife products, for the pet trade, and yes, even the bushmeat trade. So misguided hatred directed towards people of Asian origin is entirely unjustified. So what we're missing here are a few different things. Our pandemic interception and response systems are pretty slow and uneven. So for example, in September 2019, this is about 2 months before the first COVID-19 cases were reported, the USAID's PREDICT program published the results of a study that found 11 novel coronaviruses from bats in just one cave in southwestern China. And about just a little under 1% of the surrounding human populations actually had antibodies to these viruses. So this is actually not a very surprising result, because we don't know much about viruses at all when you compare the numbers that exist out there. But this would have, obviously made big headlines today, but it went largely unnoticed back then. The second thing is that the global distribution of reference laboratories doesn't actually reflect EID risk. So Southeast Asia, Africa, Central and South America, they carry the burden of EID risk from wildlife. And yet 62% of reference laboratories are actually in Europe and North America. So should we really have more labs that are able to identify new diseases? Yes, in the countries where they actually spill over to humans. The third is that the Convention on the International Trade of Endangered Species regulates trade of these species across 180 plus countries. But currently, it doesn't accommodate options to screen for disease. Adding this is underway, but imagine doing anything that 180 plus countries have to agree to. This is a slow process. And finally, we lack a blueprint of how we can adapt technology and skills to actually apply them towards disease surveillance for wildlife in situ in the countries where spillovers might happen. And we need to expand our ideas of who gets to do wildlife surveillance. So 1 year ago, you needed a medical degree to swab someone's nose, right? Today, anyone can take a swab-based test at home. So the new blueprint here has to be affordable. It has to implement a solution that is in situ, and it has to have some support from ex situ sources as needed. So what we can do really are to build better systems. Laboratories need not be located in cities. They need not cost hundreds of thousands of dollars to set up. They need not be operated by people with PhDs, and they are not by definition unsafe if they don't look like laboratories the way we think about them today. Current biosurveillance is also really centralized. It's expensive and it's maintained by a few select countries. It relies largely on exporting samples collected in countries where we think disease surveillance has to be happening, and I agree with that, but we rely on exporting these samples abroad. And when we do that, we actually export all the opportunities to participate in analyses as well. Third, large coordinated screening efforts have been funded this past year, and this is excellent news. But I think we need to diversify our approach so that political whims cannot dictate science. And one way to do this is to make laboratories both cheap and also not entirely reliant on governmental sources of funding only. So once we do this, we can actually envision a more democratic, decentralized laboratory network that cheaply can monitor wildlife populations year-round, and it should be led basically by range country scientists. So the international wildlife trade is a substantial global industry, and it's in need of greater oversight for sure. And I would refer you to this excellent editorial put together by some of our focus group members and to the sources within it for more information on how consumer-focused demand reduction efforts are actually both cheaper and more effective than other comparative methods. Third, we really have to embrace technological advancements that can reimagine laboratories. So low-cost portable technologies, particularly in the realm of molecular detection, are revolutionizing how we think about where science takes place and who can do it. Miniature genome sequencers like the Mrinalini, or tiny affordable thermocyclers, affordable automated pipetting robots like the Opentrons, these things can actually make low-cost labs be safe. We also need publicly accessible, centralized, curated repositories for pathogen data that can reflect any pathogen, right? Large numbers of things that we may not even know or have names for yet. And we need biobanking facilities that are also more widespread. For example, by associating them with all museums, that would be one way to go. So preserving tissue or DNA, I think, is the wisest investment that we can make towards the future, in which technological advancements can beggar belief today. Like we might come up with something that's completely new, and collecting those tissues right now is really, really important for us to do to be applicable then in the future. So one thing that we really have to think about, and I am absolutely owning here that I am part of this problem, is who are we really leaving out of the conversation? So the most common story I hear from people who have approached me since this perspective piece was published is actually from range country scientists or NGOs, people that work for NGOs there, who once participated in disease surveillance programs. So they assisted in what were often just one-time screens of large numbers of sites in their countries. They brought samples to a central lab that was outfitted to stabilize the samples. So doing things like converting RNA to cDNA, for example, and then the samples left the country for analysis elsewhere, and no one ever came back. These people are talented scientists. They have the skill sets and the drive and the knowledge that cannot be replaced by any number of outsiders, and they have to be a part of this conversation. I am a tad biased, I know, but I really do believe that people who work with wildlife have a perspective to give that is simply not easily found amongst those who haven't had to face the sorts of barriers that they have. So to get funded or even to obtain a precious fecal sample from some cryptic species you cannot actually follow, who won't tolerate your presence, but the fecal sample is the only way you can actually assess its health. And finally, museums and zoos hold the world's biodiversity. They save tissues and specimens in addition to saving species, and that can give us a window into the past. And these places are often the final resting place for some species that is extinct in the wild. So do I have all the answers? I think that's an emphatic no. But as a research scientist who, along with my colleagues who formulated the ideas in this paper that we put together, we work on non-model organisms in less than convenient situations. And I think our approach is different. It tends to question assumptions, and therefore it's doing what science fundamentally is supposed to do. We ask why, and then we say, why not try something different? And so for a little bit more information on the first node in a decentralized network that we're helping to set up in South America, please go to institutelabs.org, and that will give you some information there. But I'm also happy to take questions, of course, afterwards in the breakout room. And that's me. Thank you.
Excellent. Thank you so much, Mrinalini. I love the shout out to museums there, of course, at the end. We don't, unfortunately, have time for another question right now, but we'll have plenty of time for some questions in the group Q&A and then also in the breakout room. So keep them coming in the chat there. And we are going to move on to our next speaker, who is Dr. Erik Hofmeister. He is a research virologist at the USGS National Wildlife Health Center in Madison, Wisconsin, where he conducts research on West Nile virus, avian malaria, and the effect of neonicotinoids, or what people commonly refer to as neonics, insecticides. He's looking at the effect of those on wild birds. After graduating from Cornell University as a veterinarian, he practiced large animal medicine for 10 years before returning for a Ph.D. at Johns Hopkins School of Public Health and a postdoctoral fellowship at the Mayo Clinic. Erik is a real person too, with children and a priceless granddaughter, and his favorite place is the big lake, Lake Superior. Thanks, Erik, for joining us. Looking forward to your talk.
Thanks, Michael. Emma, can you go back 2 slides to the title slide? Great. So I'm going to move this talk to a slightly different place, and that would be the effect of climate change on zoonotic diseases, and use 2 examples. Just to make sure, and could you next, please? Okay. Good. Just to make sure we're all on the same footing, I'd like to briefly address the definition of zoonotic diseases. These are diseases or disease agents present in animals that also cause disease in humans. In some cases, they cause disease in animals, but in other cases, there's no apparent ill effect on the host animal. I might also mention there are what are also termed reverse zoonoses, and those are diseases of humans that manage to make their way to animals. So like I said, the 2 diseases I'll consider today are Lyme disease and West Nile virus. Next, please. Also, so we're also on the same footing. Climate change caused by the accumulation of human-produced greenhouse gases was predicted by the Intergovernmental Panel on Climate Change to result in the following. In the Northern Hemisphere, surface temperatures increase, hot days and heat indexes increase, cold days and frost days decrease, continental precipitation increases, and heavy precipitation events increase. There will be and is occurring a decreased snowpack and earlier runoff from glacier and snow-fed streams. Also the increased temperatures of lakes and rivers. Nearly all of these have been observed in North America. Next. Thank you. Epidemiologists rely on the epidemiologic triangle to relate the host, agent, and the environment. Without going into this too deeply, one can consider the impact of stress on host susceptibility, acquisition of mutations on agent virulence, and temperature acting on both hosts and agents. In terms of an agent, I'm including bacteria, viruses, parasites, and any infectious organism. If you'll just hit enter again. And once again, that's the last animation. For vector-transmitted diseases such as mosquitoes or ticks, there's the additional element of survival of the agent in the vector, and also the effect of the environmental factors on survival and distribution of the vector. Vectors include arthropods such as mosquitoes, ticks, midges, and flies. Next. As was mentioned in a previous talk, the agent of Lyme disease, Borrelia burgdorferi, is maintained in both warm-blooded rodents and to some extent in birds, and also in the tick vector, Ixodes scapularis in the east, in the mid-central part of the US, and Ixodes pacificus on the west coast. Because the bacterium does not pass from the female adult tick through the egg to larval ticks, infected rodents such as white-footed mice are essential to infect the new crop of larval ticks each spring. And one thing, if you go back just for a second, one thing I might mention there is the tick vector of Lyme disease requires a full 2 years to get from the hatching egg back to a hatching egg laid by another female. So that it's a very long life cycle for a vector. And also that it appears that with no ill effect on mice, the mice remain perpetually able to infect ticks through their lives. Okay, next. The distribution of Lyme disease in the US is primarily in the northeastern and upper mid-central states, and to some degree on the west coast of the United States. Okay, next. Ixodes scapularis ticks, the vector in the eastern US, has been spreading in part due to climate change. This is work by Nick Ogden in Canada, showing in the yellow arrow at the very bottom leftmost panel. It's a little bit hard to see, but it's Long Point Provincial Park, which extends out into Lake Erie in Ontario, Canada. That was the only spot where Ixodes scapularis was detected in 1990. The black sites, the other arrows in black show new areas of detection in Ontario, Quebec, New Brunswick, and the Maritime provinces. While translocation of the tick is likely aided by migrating birds, climate change has also brought milder winters to southern Canada. Next, please. Based on IPCC climate models for North America, through the mid-century, predicts further spread of Ixodes scapularis ticks in southern Canada, resulting in an increased risk of Lyme disease. On the upper left is the period of 2000 to 2019, and on the lower right, through the mid-century, the moderate risk of encountering population of Ixodes scapularis farther north increases as shown in the brown. The risk of migrating birds carrying ticks in their northern migration is shown in green, and you can see that the area colored in green decreases from basically the current period into the future, and it's replaced by brown, which indicates that there's population of Ixodes scapularis will likely be found in those areas. Okay, next please. Another area of Lyme disease ecology that might be affected by climate change involves the winter habitat for small mammals in the northern US. The subniveum is a thermal refugium for many small mammals in northern forests. In the subniveal space, and that's beneath the snowpack, small mammals can avoid predation and also find food in the winter. Climate change predicts declines in the extent, duration, depth, and change in the density of the snow cover. Next, please. This is a great figure by Ben Zuckerberg and John Pauly from UW here in Madison, showing the relationship between the subniveal temperatures and the snow density. So on the left, at the bottom, there's that shows basically the historical setup, which shows a space beneath the snowpack and very little heat loss through that snowpack. Then if you look in the figure in the center, it shows in blue the snow temperature and in brown the snow density. So historically, the density has been low and the temperature higher. If human activities such as snowmobiling or logging compacts the snow, so you have a loss of the subniveal space, you have a lot of heat escaping through the snowpack. And in the center panel, it shows temperature in blue fluctuating, but lower than the current historical levels. Also, the density of the snow is compacted by all the machinery. Last, the climate change has brought us a change in the type of snowfalls that we're experiencing. Wetter snows, plus also rain on snow events. And in relationship to the historical pattern, what that does is it decreases the temperature of the snowpack and also increases the density. So there's an effect of climate change on the subniveal space, even without human involvement. Next, please. The second disease is West Nile virus. West Nile virus is maintained in an enzootic cycle involving birds and bird-feeding mosquitoes. Yearly, once this cryptic cycle builds, cryptic in the sense that we aren't even aware that this is happening unless there are large birds that die that are found, such as crows and blue jays. Once this cryptic cycle builds, mosquitoes that will feed on both birds and mammals, termed bridge vectors, transmit the virus to humans and other mammals such as horses. Infected humans and horses are termed dead-end hosts because we don't develop enough viremia to infect feeding mosquitoes. However, there is rare zoonotic transmission between a mother and her fetus, also a mother and her breastfeeding child, plus there can be transmission, human-to-human transmission through transplants. Next, please. West Nile virus was introduced into the US in 1999 in the New York City area and was transmitted in the eastern seaboard states. However, in 2002 and 2003, the virus radiated to the Midwest, the Rocky Mountain states, and even as far as California. To do this, in part, it gained a mutation that allowed it to have a fast replication rate in mosquitoes. And secondly, it also encountered a second mosquito host, which is Culex tarsalis. So 2003 shown in that graph has a huge number of human cases in blue. Another year that had a large number of cases is 2012. Next, please. In 2012, transmission primarily involved California, the southern states, and the upper Midwestern states shown here as pink counties. If a person was found infected in a county, the county is painted pink by the CDC. I also looked and found that in 2012, there was also a boost in the number of cases reported in Canada, and it was probably in those prairie colonies of prairie provinces. Next, please. The possibilities for that uptick in human cases in 2012, people proposed that it may have been another mutation of West Nile virus, and that wasn't detected. The loss of specific immunity in avian hosts, termed herd immunity. We hear a lot about herd immunity now because of coronavirus. Because birds, the population of birds is added to yearly by newly hatched birds, herd immunity is dropping if there isn't very much West Nile virus transmission. So the loss of that immunity is believed to be part of the intense transmission in 2012. Drought. The US was under intense drought in 2012. What drought does is it forces a higher density of birds at existing water. It also increases mosquito populations in areas that do have some water. With drought came in higher environmental temperatures. They also increased mosquito development time, decreased mosquito development time, and also increased replication rates within the mosquitoes. Next one, please. Wisconsin also experienced a small blip in cases in 2017. In that year, there was a near record precipitation in the first half of 2017. It was the second highest recorded since precipitation has been recorded. That led to a bumper crop of mosquitoes. Madison and Milwaukee had one of 10 hottest years recorded in 2017. So there definitely is a climatic effect on the transmission and the occurrence of West Nile virus. Next, please. Taken together, precipitation and environmental temperature may have an effect on the risk of West Nile virus in the near future. This figure shows the percent change in water stress index by the mid-century compared with historic values between 1900 and 1970. Blue shows where climate change is likely to increase surface water supply, more rain. Brown means global warming and its precipitation side effects are projected to decrease the water supply, making water stress worse. Next, please. So I've considered two zoonotic diseases. In Lyme disease, interestingly, there seems to be little effect on the primary reservoir host, whereas with West Nile virus is not only a zoonotic disease that affects people, but it's killed literally thousands and thousands of birds. Also, with these two diseases, I thought it might be interesting to finalize this by thinking about the differences between ticks and mosquitoes as vectors. The life cycle, as I mentioned, for ticks takes months for stages, but actually literally 2 years to go from egg to another hatched egg. In mosquitoes, stages are accomplished in days, and it takes just weeks to months for replication to occur. So they're much better at responding to environmental perturbations. In terms of increased temperature, still within the optimal temperature range for the mosquitoes and ticks, there's faster development in both ticks and mosquitoes. For activity and mortality, there can be an either increase with low increases of temperature, but if the temperature change becomes too high, then you can see decreased activity and increased mortality for both ticks and mosquitoes. So there is definitely a thermal optimum for both of these vectors. In terms of increased humidity, which is also projected by climate change, there's increased activity in both ticks and mosquitoes. And in terms of increased rainfall, that tends to inhibit the activity of ticks, whereas it increases the reproduction and population expansion of mosquitoes. I've borrowed these ideas from Ogden and Lindsay in 2016. Next. And with that, I'll stop and take questions in the breakout room. Thank you.
That was excellent. Thank you, Eric. I love the conclusion with a little bit of entomological, or at least arthropod life history and how it pertains to all of this. Thanks for taking the time to put that story together. And as you said, we are going to move on, and people can head to the breakout rooms for questions with Eric when we get to that point. But I want to introduce our final speaker, Professor Robert Montgomery. He is a faculty member in the Department of Zoology, a senior research fellow in Lady Margaret Hall, and an associate of the Wildlife Conservation Research Unit at the University of Oxford. Currently, Bob is the managing director of the world's first ever conservation venture studio at Oxford, which seeks to rapidly mobilize tangible and technological solutions for the world's pressing environmental problems. Most recently, Bob was at Michigan State University, where he built his laboratory to research the ecology of carnivores and their prey around the world. Now at Oxford, Bob has embraced the challenge of re-engineering to better confront prevailing gaps between research and implementation so as to create a more prosperous and sustainable future. I was very excited to have Robert join us today because I predict, I'm putting on my prediction hat here, that in the coming years, we are going to see a lot of work coming out of how wildlife has responded to the COVID-19 pandemic. And Robert is one of the first people to start thinking about that and putting some ideas and hypotheses out there. So Robert, thanks for taking the time to be with us today. Really looking forward to your talk.
Well, thank you, Michael, and what an honor to be a capstone on these 4 marvelous presentations today. So it's just a great honor to be in everyone's company. Thanks to Michael, a real special thanks to Emma and to Fernanda as well, who has been working so diligently to translate this over on the Spanish channel. So thank you. I understand this is a lot of work and deeply appreciative. So as Michael well described, this paper and this idea was born out of looking out of my living room window during lockdown and wondering what the animals were doing and how they were responding. I really don't need to say very much at all about what COVID-19 has done to us all. This is a pandemic that started just over a year ago in December of 2019 in China. And given the transportation networks and corridors throughout the world, it quickly spread to the point at which by March, our lives had fundamentally changed because we were all in the midst of a pandemic. And at that time, greater than 100 countries implemented some version of a lockdown to try to control the spread of this virus, substantially changed the way that society functioned. And with that, we had to put our heads together to figure out exactly what we were going to call this period. And we decided to call it the COVID-19 quietus. Quietus is a term that has multiple definitions. If you look to the writing of Bill Shakespeare, you can see that it's a way of relinquishing debt. But we take the Oxford English Dictionary version of a calming effect, because it certainly had a calming effect on human society. So what do we know about the COVID-19 quietus? Well, fossil fuel consumption, because we stopped driving and stopped flying, reached 25-year lows, which plunged oil prices into the negative for the first time. Daily global CO2 emissions were reduced by 17%. And fundamentally, what we did and where we did it was changed, completely reshaped. And an interesting artifact of this is that many people actually began to spend more time in nature, in that they were out on hiking trails and had the binoculars out and the bird books trying to figure out what the wildlife were doing. Unfortunately, we also had a lot of misinformation in this time period. So COVID-19 actually led to a number of completely false or exaggerated stories of wildlife returning to different parts of the world, likely to bring hope to people that were suffering from the pandemic. And there was also a question about whether or not the patterns that we were observing were actually authentic or merely a byproduct of the fact that we as a human society became more observant. We weren't driving into work, we were actually working from home, and interesting things were passing our windows. Importantly, the changes to animal behavior during the COVID-19 pandemic should be expected. And as Michael articulated, we will see scores and scores of scientific studies published in very high impact factor peer-reviewed journals that are demonstrating that wildlife fundamentally did different things in the pre and post period. That is what should be expected. But why would it matter if a wildlife choose to do something different during this period? Well, it might matter because we have behavioral changes that can potentially affect survival and reproduction, both positively and negatively. Survival and reproduction at an individual level can actually scale to have population level consequences. And those population level consequences have impacts on animal conservation. But importantly, what we must do is we must search for the impacts or the costs of these behavioral changes on survival and reproduction. Otherwise, it merely demonstrates that wildlife are quite adaptable to their environment, which is what we would expect from evolutionary principles. So what we decided to do was actually to develop a topology of animal behaviors, which was basically a categorization of what we thought might be changing in animals so as to guide comparability across species and across studies of changes that we were observing in the COVID-19 quietus. We came up with 7 things that we thought might be relevant. The first is activity schedules, exactly when animals are awake and when they are asleep. We see profound global patterns, shifting of activity cycles associated with human activity, and we're wondering as to whether or not they may have changed during the pandemic. Vocalizations. There was a very nice study out of the San Francisco Bay Area with white-crowned sparrows, which demonstrated a modulation in the vocalization of those sparrows according to a reduction in the acoustic noise that was in the system. Exploratory behaviors. We have some very interesting studies emerging right now different animals that are exploring a broader part of their environment because now those roads don't have as many cars on them. And so the ways in which that might change in terms of costs and benefits, with those exploratory benefits, you can have an increase in vehicle strikes as well. Movement dynamics. This is a whole suite of ecological research focused on the ways in which animals move about their environment, looking at speed and turning angle and all sorts of things like that. Are we seeing that those variables are changing in the middle of the pandemic also? Ranging and resource use, basically animals using a broader section of their environment that they hadn't been able to use before because of profound human disturbance. Density changes. In the Yosemite Valley, as an example, we had larger aggregations of black bears than had been seen before, given that the park was closed and there were no tourists. And then modulation in the vigilance of animals. So typically animals, according to prevailing risk in their landscape, whether they be from sympatric species or from humans, have to be vigilant so as to detect those risks and then make adjustments to their behavior. Perhaps those things could have been relaxed as well. Well, the question really is, where do we go from here? Importantly, animal behavior could increase, decrease, or remain unchanged on any of these 7 categories. We're not presuming that humans are terrible for animals, and there are many animal species that actually benefit from close proximity to humans. And so it's important that these merely set up a system of hypotheses that could go out and be tested with future studies. We really must look at these patterns across sites and across species in order to understand whether or not the observations that we're detecting are replicated in different places. The other thing that I'd like to say is I'm not presuming that this period was a panacea for animals. In fact, for many animals, it was the exact opposite because we had historically low rates of tourism and decreased levels of protected area surveillance. This was a problem all over the world. We oftentimes conceptualize poaching to be a problem in the global south, but poaching and illegal animal harvest actually increased in the global north as well because surveillance was at all-time lows. The other thing is that there will be persistent and long-term negative consequences of the COVID-19 pandemic on conservation, particularly in the world's global south, because so many of our conservation funding mechanisms are predicated either directly or indirectly upon tourism. And it's currently unclear exactly when tourism will return to pre-pandemic levels, and that could be a very long-term problem. However, despite the fact that we had to implement very austere measures that many of us were directly affected financially and personally with loved ones that we lost, the COVID-19 pandemic also presents an opportunity for us to learn. It allows us to study the impacts of our collective human action on animal ecology. It allows us to understand the regulation of ecological relationships so as to inform progressive conservation activities. And the general message here is, given our austerity, we can actually become empowered by our collective decision-making to create a more prosperous and sustainable future for wildlife and for people on this planet. So with that, I'd be happy to take any questions. This is a photo from South Africa, a golf course that had not only African wild dogs, but if you turn the camera around, there were lions and spotted hyena as well. This was very early on in the lockdown measures of the pandemic. And I think all these animals had been wondering what the fuss was with these golf courses. So they decided to check it out for themselves. So thanks very much.
That's excellent. Thanks so much, Bob. And one of the study that you brought up about the white-crowned sparrows in the Bay Area, one of the authors on that paper, Jen Phillips, used to volunteer here at the San Diego Natural History Museum, and we're hoping to bring her in for a brown bag seminar in the future so we can learn more about that interesting study. I did put a link to that in the chat if people are interested in that particular study. We're going to save the questions for the whole group here in just a second, but we are now doing that moment where we give our speakers a break, we give our translator a break, so everybody can maybe grab a cup of coffee, bio break, whatever they need to do. We are going to return back here in, let's give our 6 minutes. We'll return back here at 10:10, okay? So see you in just a few, gang. I think we will go into a little panel Q&A here. And first, I just want to start off by thanking you all. This is really a fascinating collection of talks that I think, hit on a couple of different sort of sources of anxiety that the general public, I think, has about, we've felt so vulnerable, with a global pandemic. And so it touches on a little bit of that, touches on some solutions. And it really, each story by themselves was incredibly interesting, but then even threaded together, I think there was a story there. So thank you again for taking the time to join us for the State of Biodiversity. I really appreciate it. I have a question first for you, Bob, and it really stems from the introduction in your recent move to the UK from Michigan, and the idea of this conservation venture studio at Oxford. That sounds interesting, but it also sounds like hard to understand. So can you clarify for me what you guys are about and what the goals and strategy is of a conservation venture studio?
Yeah, so this was born about 18 months ago when I formed a collaborative partnership with a venture capitalist under the title of Global Accelerated Ventures that had the opinion that conservation is the next big industry, and that it's merely our preconceived notions about the ways in which we've thought about conservation as being sort of grounded community-based activities that require money rather than have any potential to generate it. But essentially what the Conservation Venture Studio does is it provides support to rapidly mobilize research and development activities necessary to get various technologies, process-oriented solutions to proof of concept phases where they can be deployed to solve real-world problems. So I'll give you an example. A certain piece of technology that was originally studied and evaluated for underground tracking of badgers. It's actually a really profound challenge because GPS and VHF telemetry technology doesn't work underground. So a computer scientist at Oxford decided that he would set up a system to study the movement of badgers underground, and that same system actually proved to be quite influential in technology that could be embedded in construction materials, namely concrete, that could detect tremors, foundational structural breaks in the structural integrity of the building, and would be able to report back to the building owner a certain section of that building that has a problem. So the idea is that you have technologies that are currently being validated for a certain purpose on university campuses that have parallel verticals that can make inroads in conservation and sustainability. And so that's just one example of dozens of technologies that we're working through right now at Oxford. And again, we have a philosophy in mind for scaling the Conservation Venture Studio to multiple university partners with time.
Cool. Great. That's interesting. We're exploring a project here at the Natural History Museum of sort of monitoring backyard biodiversity, and been looking at acoustic monitoring of birds. And when I started searching for the different potential for acoustic monitoring, it's amazing. There's all these like little Indiegogo startups of scientists who are putting together a Raspberry Pi with a microphone to try to detect these things. And so I totally see the need to bring these things together in a way that they can be more available, I would say. So cool, thanks for that. We have a question in the chat for Mrinalini, and it's, you mentioned there are quite a few in situ scientists who have done work in remote areas to collect data on a variety of things. What efforts has the San Diego Zoo Wildlife Alliance made to include in situ scientists in the work that you all are doing?
Thanks. That's a broad question, and I can answer it giving you one particular sort of example. So this idea of setting up field laboratories is pretty much the bulk of what I do for the zoo, and our intention here is very clear from the start, and that is that we do not remain involved after a period of time, right? The point is not to actually set up programs that are entirely dependent on outside funding or run by people from outside the country. And the idea is to basically do what you can to have this conversation started, to figure out where somebody is on the pipeline of, I have some sort of lab, and this is what I want to do with it, and how do we get it to the point where it can safely actually achieve that goal. And so we help with giving technological advice, with acquiring funding for some of this, and giving the sort of quote unquote sort of startup funds, I guess, to basically set these things going. But then we do a lot of other things that are, I think, pretty boring to a lot of people. So, you know, how do you actually manage data coming in? You know, what are you going to do if you now you can sequence in the middle of the jungle, but you now have gigabytes worth of sequence data, and you don't have anyone that can necessarily look at that on a computer. You don't even have the computer to be able to do that. You know, so how do you do everything that you need to for a lab offline? How do you do it on solar power only? And how do you make those pipelines start to finish, going from sample all the way to sequence and analysis, become smooth? And so those are the sorts of problems that we try to figure out. But those are not always, you know, every institute scientist has every range country scientist is at a different point on that pipeline. And so we try to kind of assess what that is, and then give them the tools to be able to do this and very clearly have an exit strategy. Because the idea is not to actually be there and do it for somebody, and that is not what they want either. So, and they're, you know, they may not have training in the way that we very typically think people have to be trained to do certain things, but it's absolutely not rocket science. And I'm always saying, you know, if you can cook, you can do genomics, and you can follow a recipe, you can do it. And so I think everybody is capable of doing this. And so I've been able to, you know, take people from I've never held a pipette to I'm loading a MinION flow cell in 2 weeks. Why not? You know, you can do these things. And so that's sort of the approach anyway.
Great. That's excellent. Thank you. And really inspiring work. In preparation for this, I did I checked out a YouTube video of that you have of your lab down in South America, and it was really cool to see the setup and folks at work. So thanks for the work you're doing. We have a question for Erik, and it says, weaving Chelsea's presentation with your own, perhaps with urbanization, brings domestic/feral species, e.g. house versus street cats. What effects are there on the introduction of domestic/feral species on the population of vector species, i.e. the rodents, the birds, and the ticks, etc.? And we'll start with you, maybe Erik, and Chelsea, if you want to chime in with any thoughts on that as well, that'd be great.
So for Lyme disease, for instance, there may be an introduction of tick populations to an urban area brought by the feral hosts. And those could potentially expose domesticated pets like dogs and cats. So there may be an issue from that direction. For West Nile virus, there could be, for instance, if the urban area was surrounded by swamp or wetlands, the primary vector there might be Culex tarsalis, whereas in the urban area, it would be a different Culex, probably Culex pipiens in the Northeast. The vector might be, there is documentation that vectors, mosquitoes, can be blown from one area to another. So there's a possibility of feral vectors, so to speak, being blown into an urban area and transmitting West Nile virus.
Yeah, I'll just jump off of Erik's excellent answer and add that it's so interesting when you delve into these individual diseases, because there are so many various ecological dynamics that could act in either direction. I think your hypothesis that street cats could exert some predation pressure on, for example, the rodent species that carry Lyme disease, and thereby reduce risk. I don't know of any empirical examples of that happening, but it's certainly plausible. And then Erik, I'm also thinking about all of Marm Kilpatrick's work on West Nile virus in Washington, DC and the suburbs outside, where he was able to show that there was this kind of hump-shaped relationship, I think, between rural and urban areas, where the more American robins you get, the more West Nile there is, because they're amazing at amplifying the virus. But you also have to coincide with the distribution of the mosquito that feeds on both birds and people. I don't remember what mosquito species it is, but it happens to be more common in kind of like peri-urban areas. And so it's this interaction of where the reservoir host is and where the vector is that determines where risk is greatest. And so understanding the like underlying ecological dynamics is really important. I'll give you one more example of the way in which introduced species might influence disease transmission. And that one comes from Palmyra Atoll, which is this tiny coral island out in the middle of the central equatorial Pacific. It was a Navy base during World War II and otherwise has been pretty much uninhabited for its entire existence. I did a little bit of work out there when I was a PhD student. And there were invasive rats on that island for many years, probably brought there by sailors before World War II. And because there were rats, there were tons of mammal-feeding mosquitoes, which made it a really uncomfortable place to work, particularly at dawn and dusk. I remember when I used to spend time out on this island, planes would come in occasionally, and we would always like rake the camp to make it look pretty for the people coming in on the planes. And that raking was torture because we would just be attacked by mosquitoes constantly. I would be covered in mosquito bites after this raking. So of course the camp would look great, but we would all look like a disaster to the new people arriving on the plane. But then they went in and totally eradicated all of those rats. And as soon as the rats were gone, the mosquitoes were gone. So introduced species can have, you know, we can't generalize about the effects that they have. They could theoretically, like these introduced cats, reduce disease transmission to people. They could theoretically, like these rats on Palmyra Atoll, increase disease risk to people. There's, you know, ecology is not physics. There aren't these F equals MA general laws. There's lots of context dependence and individual stories.
Excellent.
So,
yeah, go for it, Erik.
So your mention of American robins made me think that there could be an example of an urban animal that carries an infectious disease to a suburban or rural area. For instance, usually by late August, you stop seeing robins in suburban areas, and where they go to is wooded thickets that are toward the on the outskirts of the suburban area. So if there's been a lot of West Nile virus transmission and the birds are infected, they could potentially carry West Nile to an area where it hasn't been transmitted that summer.
Yeah, it's fascinating the dynamics of how all these things are connected together, and lots of as I always say when people ask me questions at the end of my talks, lots of future master's theses to come out of this, the hypotheses and the scholarship that you all are generating. So again, thank you for not only being with us today, but the work that you do. I have one final question for you all before we head into our breakout rooms. And it stems from the keynote for this entire series was Nancy Knowlton, who talked to us about her sort of the Earth optimism movement. And, you know, where are the success stories, what examples of nature's resiliency, but just in general, what gives us hope in times of after a year of somewhat despair in a global pandemic. And so I just starting with you, Bob, like what are some of the things that are giving you hope about biodiversity conservation moving forward, and we'll cycle from there.
Yeah, thanks, Michael. I think this is a fundamentally important point. And it's interesting when you look back at the history of conservation biology, it's a very young discipline. It only started in 1978, actually at a conference in San Diego. That was the point at which people started to be able to call themselves conservation biology. And of course, the founding of the discipline was trying to differentiate it from other similar disciplines. And the Michael Soule, who founded conservation biology, one of the things he said is that conservation is often different from parallel fields because it's oftentimes predicated upon crisis. And historically, conservation scientists have wielded crisis like a sword to try to generate conservation action. And I think what the public is now determining is that crisis is no longer an effective way to communicate the potential for conservation outcomes. And in actuality, what we need to be doing is focusing on human empowerment. So I find great hope out of collective human action and takeaway messages that we can derive from the way in which we've all sacrificed and responded to the COVID-19 period to determine exactly how we can reshape the future, again, to make it more prosperous and sustainable.
Sorry. Yeah. Yeah. Thanks for that. That's certainly a message that I think is apropos. Erik, how about yourself? What are you hopeful about other than your beautiful grandchild?
I'm hopeful that our administration is now taking climate change seriously, so that my beautiful grandchild might at some point consider having children herself, and they might consider having children, and that the world they might live in might be as nice as what we have right now.
Yeah, without a doubt. I have kids of my own and the same anxieties and hopes and dreams as well. Mrinalini, how about yourself?
I think I'm not sure I can put it terribly eloquently, but I think the thing that makes me the most hopeful after having gone through this year is seeing exactly how flexible and adaptive humans have been across the globe to being able to get through this difficulty together, and that our world in the before times was very labeled. You did this education, you were this person, you took that that step, you took that, you had this career, and now it feels much more like people's skills are being appreciated, that it's what you can do, and all of us can do so much that actually matter. And so people are being incredibly inventive, resourceful, and there's a word in Hindi that we say it's called jugaad, and it's that idea. It's what field biologists are doing all the time, because nothing's ever designed for what you need, and you take one thing and apply it to another purpose, and it's sort of that in itself is being appreciated, and it's being explored so much. And I think that to me just gives me hope for the future, because we can't predict what the next outbreak will be like, and we don't necessarily know that it'll be anything like this particular one. I mean, it could be a completely different disease with a completely different dynamic, right? And the fact is that I think that I mean, there's lots of frustrations, but I think overall I'm hopeful about the future because of that.
Yeah, awesome, thank you. And Chelsea?
Yeah, I'll tell you, one of the things that surprised me most about the project that I discussed today was that when we compared the burden of disease between 2010 and 1990, it had actually gone down. So for the 24 most important infectious diseases of people, we're actually making progress against them. We have to leave aside COVID-19 entirely, because that is a whole different story. But I was really surprised to see that we're actually making real progress on diseases that are just at shameful levels of burden in the global South. I think the other really hopeful thing that came out of that analysis was that urbanization and wealth appear to have this protective effect, and countries in the global South are rapidly urbanizing and they're becoming wealthier. And those both portend good things for the transmission of those infectious diseases. So I think those were really hopeful outcomes and give me kind of faith in the fact that we're making progress against these diseases that have no business still affecting people.
Yeah, I think so much of there have been the reports that have come out in the last couple of years about the state of biodiversity from the ICEP, but and I know and just in general what we're seeing, I think, is more of a conversation about like including indigenous knowledge and local expertise and people on the ground in those areas as part of solving these big issues. And I think I see just some parallelisms between Mrinalini's work and like in yours, Chelsea, in the sense of like that some of those same things could be put into effect in the global South in areas to help with monitoring and disease control down there. So anyways, that's just me rambling on. I want to finish off by saying what I'm most hopeful for, and what I'm and I said this to the last group. I won't lie to you, but it's that you guys give me hope that there are people like you working out in the world to really improve the state of biodiversity, to increase the scientific knowledge, to question our expectations, subvert our expectations of what the world really looks like, so that we can, understand things better and make data-driven decisions, while also sort of respecting the local expertise of the areas in which we're working. So thank you all for being with us today.